Thank you very much for having me here today. I'm really excited to tell you today about integrating mesoscale human brain measurements in the human visual pathways. Because OHBM in 2008 was the first conference I ever attended, and it's an honor to give this lecture today. In 2008, I remember that I loved seeing all the different human brain measurements, and I just wanted to understand how we can integrate these different measurements to inform about both typical and atypical processing. And let me start by giving you an example. Because in photosensitive epilepsy, not only flashing lights can cause seizures, but also patterns of gradings and stripes can trigger seizures in photosensitive epilepsy. And the Epilepsy Foundation even has warnings about this on its website for patients with photosensitive epilepsy, because Venetian blinds and radiators can, for example, be provocative in these patients. And there are established broadcasting guidelines about what can be shown on a TV screen. And these guidelines state, for example, that more than 5 stationary stripes over more than 40% of the screen can be provocative and should be avoided in any type of broadcasting. And if these stripes oscillate, they shouldn't cover more than 25% of the screen. But if they smoothly flow, then they are exempt. And these types of patterns can not only cause seizures in photosensitive epilepsy, but can also cause headaches in patients with migraine, and can even cause discomfort in perfectly healthy individuals. And you can see here that in the Museum of Illusions, there's a room full of stripes, and it even has a warning sign that dizziness may occur. However, there is no animal model for photosensitive epilepsy that matches the human phenotype. So what my lab is working on is to be able to understand how we can integrate human brain measurements to better understand diseases like this. And our typical approach is to directly compare measurements, because we think about these measurements in terms of their temporal resolution, as you can see here on the x-axis, and in terms of their spatial resolution, as you can see on the y-axis. And for example, the EEG and MEG have an excellent temporal resolution on the order of milliseconds, but only have a spatial resolution on the order of centimeters. Intracranial EEG measurements, where electrodes are implanted on the surface of the human brain in ECOG, or at depth with stereo EEG, still have a really good temporal resolution of the order of milliseconds and have an excellent spatial resolution as well on the order of millimeters. But they measure sparsely in select number of brain regions. Local field potentials and single units, of course, measure at the level of layers and single neurons, but also measure sparsely in a few select areas. Whereas fMRI can measure the entire brain with a millimeter spatial resolution, but lacks temporal resolution. So we compare these measurements in order to understand how each of these is related. And these comparisons have taught us a lot. However, in photosensitive epilepsy, evoked potentials and neuronal synchrony as measured with oscillations and fMRI signals all increase in amplitude. So these direct comparisons don't always help us understand diseases. So what I would like to do today is to provide a different framework where we don't do direct comparisons between measurements, but we think about what these measurements encode in terms of stimulus properties, and particularly in terms of visual input is what I'll be showing, and in terms of how these measurements integrate neuronal activity underneath the electrode or within the voxel measured with fMRI. And I will focus on that for both intracranial EEG and fMRI. And in the last session, the third part of my talk, I would like to discuss how we can work together and practically and collaboratively integrate multimodal imaging data, both within and between labs, And we've been working on the brain imaging data structure and extended that to intracranial EEG measurements to make sure that we can integrate these measurements throughout many different modalities. So let me first talk for a little bit about intracranial EEG measurements, because we do these measurements in patients with intractable epilepsy, and these electrodes are implanted for the clinical purpose of localizing the seizure foci and also localizing critical brain functions. And here on this picture, you can see a set of electrocorticography ECOG electrodes implanted on the brain surface in these white dots. And you can see that these electrodes are implanted in a large number of regions because the seizure focus is generally a small area. And we think that in the other areas, we can measure typical brain function. And we have IRB approval where we can ask these patients to participate in, for example, visual perception tasks during their clinical stay. And you can see in this case that there are some electrodes implanted in particular on early visual areas in the back of the brain. And when we think about these signals, there are many different features that we can extract from those. And those features tell us something different about the underlying neuronal population. And some of these features have received a lot of attention. For example, in the 1980s, people described narrowband gamma oscillations that are narrowband oscillations between 30 and 80 hertz, and that have a particular time scale in the measured potential. And if we look at the power spectrum from one of such traces, and we look at frequency on the x-axis and power on the y-axis, and this power spectral density, these have a peak in the spectrum at a particular frequency between 30 and 80 hertz. And these type of gamma oscillations have been related to underlying neuronal synchrony, the neurons doing the same thing at the same time with a particular timescale, and particularly an excitatory inhibitory circuit. And one of the reasons why these gamma oscillations received so much attention is because they are proposed to play an important role in visual perception and feed-forward processing. In 2007, however, around the early 2000s, people discovered that it's not just always an oscillation in these field potentials, and there are broadband changes that span many frequencies and that are expressed as a random walk in the raw potential. And when we look at these type of processes in a spectral power density plot with frequency on the x-axis and power on the y-axis, we see that there's a 1/f distribution. And this can increase and decrease in a broad range of frequencies. And we have related those both in modeling. We have seen those being related both in modeling work by Kai Miller and others, and in measurements as well, where they have been related to Poisson distributed input firing. And people haven't always referred to this type of process as broadband. It's also known as broadband gamma or high frequency broadband. Now, while there was a lot of attention to narrowband gamma, we often didn't measure this in human ECOG. So if we look at these local field potentials in macaque V1, papers published these very clear narrowband gamma oscillations at around 50 hertz, with a particular peak in the frequency. Here, you can see that here for this particular study. And they sometimes also reported broadband increases. But when we did our human ECOG measurements, either in motor cortex, as you can see here on the top, and we look at frequency and power during baseline in black and during hand movement in red, in green, or in ventral temporal cortex, where we see baseline in black and during phasers or heart processes in pink or blue, we did not observe these clear gamma oscillations. But we did observe very robust broadband increases. So there were many questions that we could ask from this. Do our measurements not measure gamma oscillations? People would tell us that's just what gamma looks like in human ECOG, but it's actually quite different. This has a peak in the spectrum, and this is a broadband feature. So we looked at many of the different studies that studied gamma oscillations. And what we noticed in these studies is that many of those used gratings. So we had a few patients where we had early visual electrodes implanted in patients with epilepsy, and we showed them a grating. And here you can see a single trial. And you can see a clear ERP evoked by this particular stimulus. And then a gamma oscillation visible on a single trial basis in human ECOG recordings. And when we look at the power spectrum, you can see frequency on the x-axis and power on the y-axis. And the dotted line is baseline. You see a very clear peak during this stimulus, a very clear increase. As I point to here, in red to the narrowband gamma oscillation, and in blue to the broadband increase. And we also showed other contrast patterns, noisy patterns. But what we saw for those patterns is we saw a clear broadband increase and no oscillation, and only this broad increase that spans many frequencies. And I want to look at these spectra in a little bit more detail, because what you can see here is that this gamma range between 30 and 80 or 50 and 80 hertz is one band, but has multiple processes. And the classical approach that we take to analyzing spectral power changes, where we look at a particular frequency band and link that to some kind of input, will not work in this case because one frequency range has multiple overlapping processes. And one of the other things that's important to consider is that oscillations are defined by a peak frequency, bandwidth, and power, as has been established for a long time by Fernando Lopes da Silva and Klimesh already in the 1990s. And according to this definition, this is a gamma oscillation. This is not an oscillation. So we have to separate these two different processes, and we cannot just take a particular frequency band and link that to one process. So let's first look at a couple more of these electrodes and what type of stimuli we used, because these findings were extremely robust across many different stimuli. Gratings with different spatial frequencies, a plaid, and different noise patterns. And we see that in these gratings, early visual cortex showed a very clear narrowband gamma oscillation. Whereas if we look at all stimuli and the noise patterns, they showed a very clear broadband elevation. And for each of these stimuli, we decomposed the spectral power into these separate components and assigned and fit a line plus a Gaussian to assign a weight to the narrowband gamma oscillation and a weight to the broadband changes. And I really appreciate the similar methods that have been developed by Brad Wojtak's lab to parameterize power spectra in different ways. And when we do this, when we separate the signal into a narrowband and a broadband component, we can look at the spatial distribution of the narrowband, for example, across many of these electrodes implanted in V1, V2, V3, HV4, and VO. And we see that the narrowband gamma oscillations are robust and widespread across visual cortex. Whereas electrodes didn't show significant narrowband gamma for noise patterns. And if we look at the broadband increases, we also see that in a widespread manner across many visual areas, but more specific to function. And I would like to point out that the fovea here on the tip of the back of the brain shows clear increases in broadband for high spatial frequencies, whereas it doesn't for lower spatial frequencies, and the periphery shows more increases, which matches what we know about visual function. And one of the questions that you could ask here is, this is just a noise pattern. It doesn't have any content. So what about other stimuli? If we actually have stimuli that have content, such as two different cars. We see a clear broadband increase in power, as you see here in red, compared to the baseline in gray for both of these scores, but only one of those shows a robust, reliable gamma oscillation for this type of stimuli. And the level of broadband and the level of gamma for this particular stimulus was really reliable, was reproducible on a trial-by-trial basis. But what makes it such that this scar induces a gamma oscillation and this one doesn't? So we've seen that ECOG can measure narrowband gamma oscillations. And we've developed methods to characterize and parameterize the power spectra. And these are essential to be able to distinguish narrowband from broadband changes. And gamma oscillations have different stimulus specificity than broadband responses. And we're not the only group showing this. There have been several other groups that have reproduced and extended these findings in several different ways. And this is consistent with smaller measurement scales. For example, with LFP recordings in macaque, and it shows a similar stimulus specificity. So these changes that we report are not just due to the fact that we record with an ECOG electrode rather than a local field potential. And you may ask, this changes our idea of what gamma oscillations, what the function that they may have for visual processing. Can you reproduce these results? And yes, you can reproduce these results. All the code to reproduce these figures is available on GitHub with links to the data repositories. And this will be the case for every single result that has been published that I show in this talk. And so the question that I would like to ask here is not whether there is any narrowband gamma for certain stimuli. The question that is important is how we can explain the observed variability across these stimuli in the data. And for that, we have to consider what each type of measurement encodes. And we can build on image computable encoding models that have been developed for functional MRI. These models take a stimulus, simulate a network response, and then predicts a particular level of a response. And for example, Kendrick Kay and colleagues developed such a model to predict fMRI bolt signal changes. And they filter the stimuli with Gabor filters to compute oriented contrast energy for different orientation, and apply divisive normalization and summation across orientation to calculate a contrast image. Then estimate the population receptive field with a 2D Gaussian and apply several nonlinearities to predict the response measured in fMRI. And they used a large set of 86 different stimuli that vary across space. We can here see these complex curvy patterns that cover different parts of the visual field. Gradings of different orientations. Gradings of increasing contrast, blats of increasing contrast, 8 orientation stimuli of increasing contrast, and these curvy patterns of increasing contrast. Stimuli changing in sparseness, coherence, and noise. And we presented these stimuli to a number of ECOG subjects. So these are stationary stimuli presented for 500 milliseconds to 3 subjects that had electrodes on V1, V2, V3, and some other early visual areas. And we see here the baseline in black for 3 example electrodes, the response to noise in blue, and the response to a grading stimuli in red. And we again parameterize this power spectral density with a broadband increase and a narrowband gamma increase. And we can compare that to the fMRI measurements from Kendrick that he did before in these stimuli. So what you see here on the x-axis is for every single stimulus, the level of bolt increase on the y-axis. So you can see that these type of stimuli elicited a larger bolt response compared to gratings. You can also see that the response increases with contrast, increasing contrast. And when we look at one example electrode that was representative of all the electrodes on V1, V2, and V3 that we measured, and we extract the ECOG broadband component for each stimulus, you can see that this has a very similar pattern as fMRI, with these responses to complex patterns larger than a response to gratings and increasing response for increasing contrast. So we can use this image computable model that was developed for fMRI to predict the ECOG broadband response. And across these electrodes, we could explain 80% of variance cross-validated. So ECOG broadband encodes similar visual features as fMRI. However, when we look at ECOG narrowband gamma for each stimulus, it has a very different response pattern, with responses to gratings larger than these more complex patterns, but also an increase for contrast, but not for these type of patterns. So we had to develop a different image computable encoding model to explain what type of image features predict gamma oscillations. So the first step in this image computable encoding model is similar as what was used for fMRI up to our calculation of the oriented contrast energy images. But then we maintain the different orientations, integrate that across a population receptive field. But for each orientation, we have these summed orientation contrast energy, and we can calculate the variance across those orientations in order to predict the response. So orientation variance varies with how many orientations are present in the population receptive field and with contrast. So let me show that a little bit in a little bit more detail. So in this orientation variance model, we have, in this case, if we imagine a very large population receptive field, you can see that this horizontal orientation has a large contrast, whereas the vertical orientation is not present. And if we calculate the variance across these values, we can get a certain value, in this case of 0.06. Now, if we reduce the contrast, you can see that the contrast in this particular orientation decreases, and that also reduces the variance. If we have two orientations, horizontal and vertical, that are present, so vertical has contrast, horizontal has contrast, that also reduces the variance compared to a single orientation. And especially if we have a complex pattern with many curves, all orientations will have some weight, and that lowers the variance as well. So this model would predict a high value for an stimulus with a single orientation and a low value for an image with many orientations, and also reduce its output with lower contrast. So when we tested this model across to explain these gamma power for the different stimuli, we could explain 75% across the different stimuli, again in a cross-validated manner. And you can use this model for other stimuli as well. We did Lee-1-out cross-validation, so we use a training set and predict for a new stimulus what the predicted gamma oscillation, gamma power would be. And you can take it and use it in cases of many different other stimuli to test how far it will hold up, or whether we need to consider additional features to explain the changes in gamma power. And that's important because what we asked is, can we explain the observed variability in data? And image computable encoding models make quantitative, testable, and interpretable predictions and can be extended to other settings. And what we see in this model is that orientation variance and contrast in the population receptive field predict gamma amplitude. And that many stimuli that we can clearly see and observe, and that result in feed-forward processing along the visual pathways, have a lower gamma And these features, orientation variance, contrast, larger stimuli as well, have been associated with gain control or suppression in neuronal circuits. And that's quite a different interpretation for gamma oscillations and what has been proposed in the literature. And that's important because we first took this typical approach where we compared different stimuli, different measurements just in terms of their features. And now we ask the question about what do human brain measurements encode? And I want to come back to this example that I showed in the beginning of my talk, when we just compare and contrast different measurements. In photosensitive epilepsy, we learned that evoked potential, neuronal synchrony, and fMRI signals all increase in amplitude. But let's now take this approach, given what we've learned about image computable encoding models, and apply that and see how different features change as a function of stimuli. And in order to do that, we did a literature review about the last couple of decades that measured, for example, gamma power in local field potentials in MEG or in EEG for different stimulus features. And we've seen, as I showed you before, that gamma power is reduced when we increase the level of noise on a grading. So for noise masking, gamma power is reduced. And next to that, we reviewed the EEG literature where neurologists measured the number of epileptiform responses induced by different types of visual patterns. And these studies also found that the number of epileptiform responses is reduced with noise compared to a grading. And we looked at as many different types of visual input as we could find. For example, gradings of increasing size, increases in contrast, number of orientations, luminance to red-green chromatic, low to high, middle spatial frequencies or middle to high spatial frequencies, and also grading edge. And for these types of stimuli, previous studies had recorded particular increases or decreases in gamma power. And epileptiform responses by these other studies change in a very similar way, increases in size, increases in contrast, decreasing for multiple orientations, etc. However, when we compare that to firing rates for similar stimuli, we saw that they changed in a very different manner, as well as the fMRI signal. So when we ask this question about what human brain measurements encode, rather than just comparing our measurements, we see that not all stimuli that increase neuronal firing or the fMRI bolt signal elicit seizures, but particularly those stimuli that induce narrowband gamma oscillations elicit seizures. And we can hypothesize from these findings that gamma oscillations and epileptiform responses in photosensitive epilepsy are driven by the same circuit. So let's think about this a little bit further. If this is really the case, do the same stimuli that induce narrowband gamma oscillations also really elicit if we were to measure it in the same patient? And so we had a very rare case of intracranial EEG recordings in a patient that was photosensitive also to patterns. And as you can see here, in both the left and the right visual cortex, there were several stereo EEG electrodes implanted. And we showed this patient the type of stimuli that we used before. And when you see all the electrodes here, all the signals from all the different electrodes for a couple of seconds, and we show these complex patterns, the EEG was relatively quiet. But when we showed this patient a grading, within 500 milliseconds after the onset of the grading, we can see this epileptiform response evolving. And now patients have that sometimes randomly. So what we did is we counted the number of epileptiform responses after each stimulus. And we compare that to the typical broadband and gamma power in ECOG without photosensitive epilepsy that I showed you before. So this is just exactly what I showed you before, typical broadband and gamma power for all these 86 different stimuli in patients without photosensitive epilepsy. And now we counted the number of epileptiform responses after each stimulus. And we see that it is the same stimuli that drive gamma oscillations that also induce epileptiform responses. It's the exact same set. And in other cases, they're only rarely observed. And this actually indicates that we can now use this model that we developed to predict gamma oscillations to also potentially predict how provocative certain visual input is in photosensitive epilepsy. And this shows that in an iPhone app. What you see here on the screen is the screen of an iPhone, of a smartphone. And that analyzes the camera input in little squares, and it gives the output of our image computable encoding model for gamma oscillations. And when it's green, it's relatively low, and when it's yellow or red, it's a larger output. So when this camera looks around a certain environment, you can see that for certain type of areas, there is a larger predicted gamma oscillation. And escalators are especially well known to be potentially provocative in epilepsy. And patients with migraine sometimes complain about those. And also other patterns in the environment are well detected. So we can use these image computable encoding models to predict what type of natural stimuli may induce gamma oscillations and also may be potentially provocative in photosensitive epilepsy. So we've seen how different measurements encode different image properties and how we can develop models for that can now be extended to other image properties. And the next question that I would like to ask, because we've seen that these intracranial EEG and fMRI measurements differ, why do they differ? And the question that I want to ask here is, how do these measurements integrate neuronal activity? And for that, I want to show a simplified, but hopefully useful computational framework. Because we think about our different measurements, and each measurement measures from a particular neuronal population. In the case of ECOG and fMRI, they're both on the order of millimeters. And each of these measurements integrates the neuronal activity underneath the electrode or within the voxel in a different manner. And let's consider two neurons that each have the same orientation and the same distance to an electrode. And let's consider their membrane currents for these two neurons. Then we could, in order to estimate the ECOG signal, we could sum these electrodes and calculate the sum of the time series to estimate what we would measure with ECOG. But if we were to consider what type of volt response these different membrane currents would induce, we could, for example, think about how much they each vary from a resting potential. We could calculate the power. And for example, each of these membrane currents would demand one energy. We could sum that and take the sum of the power to think about the fMRI signal. And what's obvious from this simple example is that when we change the phase of one of these two neurons and change the synchrony between the two neurons, it has a large effect on the ECOG signal, but not on fMRI. See here, basically zeroes out. And this framework is likely not an accurate description of the underlying biology. But what we want to do with these models is provide a hopefully useful computational framework that help us think in a quantitative manner about our different signals and that make predictions for what we can measure with each of these. So how do we test this framework? And we wanted to empirically test it. So we needed an ECOG experiment with broadband, gamma, and low frequency power changes, simulate membrane currents that produce these 3 different signals based on existing hypotheses, and fit that to ECOG data by taking the sum of the time series. And then we could test whether we could also predict measured volt signals. And so let's start with this ECOG experiment that induces high frequency broadband, narrowband, gamma, and low frequency power changes. So low frequency power typically decreases compared to baseline, and broadband and narrowband gamma typically increase. And these 3 features have all been correlated with volt increases. And so we have this experiment where we showed the patients blank stimuli gratings and noise patterns, and we found that in this experiment, we see broadband changes, narrowband gamma changes, and low frequency power decreases that are different for the different stimuli. So we can dissociate these 3 features. And importantly, broadband has been related to Poisson input firing rates, because we have to simulate membrane currents that produce these signals based on existing hypotheses and fit these to ECOG data. So what kind of existing hypotheses are there for these 3 phenomena? And broadband is linked to Poisson input firing, and gamma has been related to neuronal synchrony. Whereas low frequency alpha changes that negatively correlate with volt have been related to pulsed inhibition. And so we simulated a population of N neurons that receive 3 different inputs. And every neuron in cortex has about 1,000 synapses. So it's likely that there are many different types of input arriving at every single neuron. And here we have a broadband input that is random, asynchronous across neurons, and has a small offset. A narrowband signal between 50 and 60 hertz that is synchronous between the neurons. And an alpha oscillation between 9 and 12 hertz that is asymmetric, pushing the membrane potential back to baseline. And we sum these inputs and apply a leaky integration to simulate the dendritic currents in each of these neurons. And then in order to simulate an ECOG signal, we sum the time series. And here you can see the simulated power spectrum amplitudes fit to ECOG data, where there's a simulated baseline condition in black and a simulated stimulus condition in red, with a certain level of broadband increase, a certain level of narrowband gamma increase, and a certain level of alpha decrease. And again, methods to characterize and parameterize the power spectrum are essential here to fit each of these components with our simulated signal. So we have an ECOG experiment with these 3 features, and we can simulate membrane currents that produce these signals based on existing hypotheses and fit these to ECOG data. And so we tested whether with these simulated membrane currents, whether they could also predict volt signals. And we took the sum of the power across these membrane currents for different electrodes, and we found that for each stimulus, the simulated volt predicted the measured volt. And in V1, we could explain about 78% of variance. Moreover, these simulations showed that ECOG gamma could be large, but did not predict volt. And that when we tried different hypotheses in step 2 for different signals being generated by different underlying activity, the data fit less well. And this is a paradigm tested for a particular setting in visual cortex. So I want to go through what this framework would predict for other paradigms. And if we first look at that in a set of equations, and I'll show it next in a set of graphs. If we think of ECOG power as a function of the membrane currents XYT as a function of T, we can equate that to the power in an ECOG signal, whereas fMRI was simulated by the sum of the power. And these two equations, the power of the sum and the sum of the power, are very nicely related in a mathematical way because the power of the sum is equal to the sum of the power plus the sum of the cross power. And this shows, and the sum of the cross power is largely driven by the synchrony between neurons. And this shows how ECOG is driven by both synchrony and activity, whereas fMRI is not sensitive to neuronal synchrony. And whenever this green term is large, these two signals will differentiate. And because this framework makes important predictions about other brain functions, let's also try to look at this not just in equations, but in graphs. So here we plot the power of the sum that we use to simulate the total ECOG power as a function of, for example, the variance between neurons, because increasing the neurons, membrane currents may increase in variance when there is an increase in Poisson input firing rates. And then let's also look at the sum of the power as a function of increasing variance and the cross power that's related to neuronal synchrony. And so if we only increase the variance in a population of neurons, we see that the power of the sum increases and the sum of the power increases, and these two will be well correlated because this green term stays small. Now let's look at this as a function of synchrony, where we have a population of neurons that's either non-synchronous or synchronous. And let's increase the synchrony across neurons and see what happens to our simulated ECOG power and our simulated BOLD. And what you see here is that with increasing synchrony, the power of the sum to simulate ECOG power increases, whereas BOLD is not sensitive to that. And that is because the cross power increases, and that influences this signal in particular. Now we have those two parameters of synchrony and activity, and we want to plot that in a two-dimensional plane, where we can increase levels of variance and synchrony between neurons, because this is what happens in a large population. Under every electrode, there's about 500,000 neurons that show varying levels in Poisson input firing rates that change the variance and different level of synchrony between neurons. So let's first consider the example that I showed before in a one-dimensional plane, in a two-dimensional plane, where we increase the variance within neurons. You can see that in this case, because this is just increasing variance and we're not traveling in the direction of synchrony, these two are well correlated. And this is what we observe if we think that the broadband signal we measure with ECOG is related to Poisson input firing. BOLD has been correlated well with broadband in motor cortex, in visual cortex, in language regions, in ventral temporal regions. So we see that BOLD correlates with broadband, and that may be the case because synchrony does not play a large role in this particular signal. But even there are many cases where we could think about increasing variance will also increase synchrony across neurons. And in this case, all these simulated signals increase. We increase both synchrony and variance. And this may be the case when we have gratings of increasing contrast, and those have been reported to induce an increase in BOLD and an increase in gamma power, and these correlate with each other. But it's also important to consider cases where we increase the variance within neurons while decreasing synchrony across neurons. And these are the cases when we get dissociations, because with decreasing synchrony but increasing variance, our simulated ECOG power decreases, whereas our BOLD increases from light blue to yellow, and this decreases from light blue to dark blue because the cross power decreases, and that has such a large influence on this signal. And we can see this also in measurements where BOLD does not correlate with gamma power for noise masking of gratings, for example. And Suresh Mithuparamaswamy has shown many examples when BOLD does not correlate with gamma. And this example shows that whenever we move along the axis of synchrony here, this green term increases. And in these cases, bold and field potential measurements can dissociate. Now I have one last example in this two-dimensional plot that is important to consider because we also see low frequency power changes often correlated with bold. And that is when we change the mean of the membrane current within neurons. It can, for example, be driven by subthreshold membrane fluctuations. And that changes the entire level of these two planes, as you can here see compared to the previous solid fields. And for example, we can see here that these increases in mean can have a large effect on all these signals. And it is important because alpha oscillations have been proposed to be asymmetric, changing the level of the membrane current, and these correlate with small bolding changes. Small bold changes. And this has been shown in many different studies, again, in visual cortex, motor cortex, language areas. So let's summarize these findings. So we've seen that correlations between measurements do not imply causal relations because our measurements encode fundamentally different aspects of neuronal processing. And then different signal features are largely driven by activity rather than by synchrony. So for example, broadband being driven by asynchronous activity, then fMRI and ECOG signals correlate well, and we can even use functional MRI to predict what we can measure with intracranial EEG. However, when neuronal synchrony plays a large role in generating different signals, so for example, in the case of narrowband gamma oscillations or in the case of evoked potentials, then intracranial EEG recordings and fMRI signals can differ. And it's important to consider that shifts in the overall excitability, as has been hypothesized with alpha power, also affect fMRI. And we need to take these variables of activity and variance, synchrony and overall mean into account when studying different neurological or neuropsychiatric diseases. So we've seen now how these different examples of how we can think about what these measurements encode in terms of image properties and in terms of neuronal activity. And in order to advance these type of studies, we have to practically and collaboratively integrate multimodal data within and between labs, because while I'm interested in intracranial EEG and fMRI, I typically don't do MEG measurements or EEG measurements, and we need to have a common framework where we store our data to be able to make these advances. So how do we integrate multimodal data practically? And I was involved in a project with Natalia Petridou and John Winnower, where we were integrating fMRI, intracranial EEG, MEG, and unit recordings. So we were looking how we could integrate that, and we found that at that point in time that there was the brain imaging data structure developed for fMRI. And it's a shared data structure for neuroimaging data. And all metadata are both machine and human readable, which is extremely important if you work with clinical intracranial EEG data and you get human readable data. You need to have human readable data. So I talked to Chris Gogolewski, who had developed the brain imaging data structure for fMRI and had made this big effort to standardize that. And he explained to me, like, this is a community effort. And there's currently an extension developed by Yomar Niso for MEG bits. And if you want, you can extend it to intracranial EEG as well. And a week later, he told me there's this guy from Berkeley, Chris Holdgraf, that is interested in that as well. And so we got together with a large group of other people to extend the brain imaging data structure to intracranial EEG data. And during this process, we closely collaborated with the EEG bits extension to make sure that we match, for example, how sampling frequency is recorded. And it's the same in these structures. And there's currently also extensions for multi-electrode array recordings. So if you want to contribute to BIDS, this is a community effort. It is open to extensions. So we've thought about what human brain measurements encode and how we can store these data practically. So I would like to summarize these findings. We have seen that parameterizing power spectrum and considering what is encoded in different signals is important. And both we can do this in terms of stimulus properties and in terms of neuronal activity. And when we do this with image computable models, they provide testable predictions about what type of visual information is encoded in different signals. And we can test those in many different other settings. And direct correlations between signals help us map this multimodal parameter space of our different signals that I showed you. And we need theoretical models of how fMRI and intracranial EEG integrate membrane currents to help understand our measurements. And these types of approaches where we ask what is encoded in each signal can help identify circuits involved in neurological diseases and help us develop new hypotheses that are testable, such as that the circuit that drives gamma oscillations is also involved in photosensitive epilepsy. And we need to share data when possible to further understand what each signal encode, because sharing code can test and advance these models. And all the data and code that I presented today was made available on GitHub with publications. So you can test it and see when it breaks, see when it holds up, see when it fails. And that's important to advance the field. Current work in my lab involves understanding what these different measurements encode in terms of neuronal activity. And we drive this by different visual input. And rather than going directly from visual input to our measurements, we also want to understand how visual input drives neuronal populations and then how we can decode these different signals. And the same for electrical stimulation. And this is my team at Mayo that we've been setting up over the last year. And I hope that you'll see more of them in the future. And I would like to thank my collaborators and the IEEG teams at Mayo, Utrecht, and Stanford that have significantly contributed to all these studies. And especially John Winnemer, who has contributed a lot to this work and was my postdoc mentor when I had 2 small kids at home, and who was always supportive in these circumstances, as well as my current collaborators at Mayo who are helping me develop these measurements here. My collaborators in Utrecht and Stanford, of course. Thank you.